I have a little bit of um, a small square on my right, so I can't see everyone, but if you have any questions in the middle, feel free to stop me. And okay. All right. So I hope that all of you have all the slides, the code for today in the form of the markdown and the data file that I shared ready with you. We will move at a pretty moderate pace, and if you're an experienced R user, you're free to move along through the R markdown and go ahead and finish it and then just listen to the presentation. Or newer users can go on doing it with me and explore the markdown at your own pace. And please ask questions in every section. And let's get started. So today we'll be doing some preliminary data analysis using the tool called Monocle 3. This tool performs both cell type as well as trajectory analysis, and also helps with differential expression analysis. It is also, it works well on both small as well as large data sets with high complexities, whether it is from the smallest NextSeq run to the biggest S4 NovaSeq run with billions of reads. This package is constantly undergoing improvements, so help us in its development. And if you use it and find that there are bugs, or you can't seem to understand some part of the documentation, please open issues so that we can make the tool much more user-friendly. A few points to note for those of you that have already used Monocle 3 in the past. Monocle 3 is a fully independent new tool. There was Monocle 2 in the past, which is no more being used. So if you're using Monocle 3, it's completely new, and you'd have to install it from scratch. And like I said, please submit bug reports if you're going to use it. There is a open source GitHub page for the tool, and there is also a Google group where you can just ask questions about the different functions within Monocle 3 if you have any trouble at all running any part of the functions within the tool. So the three main things that Monocle 3 does is cluster analysis, differential expression analysis, and trajectory analysis. And today we're going to sort of focus on the cluster analysis. And in the first half of next year, I'm going to have 2 more tutorials that'll sort of continue from cluster analysis into differential expression analysis methods and trajectory analysis as well. So if you continue to follow these series of tutorials, you're going to have one step up each time you attend a tutorial and learn something a little more complicated than the first time. So the general workflow of Monocle is this. So today we're going to start with a single cell data set, which is that one cell data set object that I shared with you. The reason why I share just one sample for these tutorials is for the sake of time, because really large data sets take a huge time to process, and we would just be waiting and waiting. So just to show you how the general pipeline works, a small sample, I think, is a good example. So we'll be starting with that sample and first pre-process it, which includes normalizing it and removing any batch effects. Batch effects come into picture when you have samples coming from different runs and you want to remove any batch-wise bias. The next step is nonlinear dimensionality reduction, in which we make either a t-SNE plot or a UMAP. The next step is to cluster the cells. Cells with similar expression are going to cluster together. And then the things that you can do after clustering the cells sort of branch out in different paths. So one of the things that you can do is compare the different clusters and identify top markers or top marker genes among the clusters. So today we are going to do that. We're going to see how many clusters we see and what are the topmost expressed genes in each cluster, and see if those genes can tell us something about what the cell type is, what the pathways are, and two more things that you can do, the two more major things that you can do are differential expression analysis or trajectory analysis, which would come in the later tutorials. Because each one is a very big part, so it requires the whole 2 hours. So um, I foresee having the next one for differential expression analysis and the one after that for trajectory analysis. And I have included links to the Monocle 3 website, the GitHub page, and the Google group in case you have questions or you want to open issues. All right, so the basic process that Monocle 3 follows is first pre-processing the CDS. So this is this noisy single cell data set that we have. The first step is to reduce its dimension. That is sort of get some structure in the data to make meaning, biological meaning out of it. Next is to cluster the data to see how the different cells are grouping together based on the similarity of their expression. Next is to learn graph. That is how along a group of cells is the expression changing from one point to the other. Learn graph is what we do when we're trying to see dynamic change in gene expression along a path or along a trajectory. So this is a very important component of the trajectory analysis. Then we plot points along the dynamic changes that are happening along a path and see what kind of genes are working together as a module, and see how even a module changes in expression as you move from point A to point B. And then you can compare two trajectories. So you can see if you start from a certain group of cells and they branch off onto like 3 different paths, then how the gene expression changes with cells branching off into different paths. So today's agenda is dealing with the first and the most preliminary things when you have single cell data. So first is loading of the cell data set, which is the primary object that will house your single cell data, and that will be used as a raw material for the entire pipeline. So we will learn how to load that, how to QC the data, how to do the initial filtering of the data, so that you're sure that you're working with confident data. And then we will also learn how to make this cell data set from scratch if you don't have one ready already. We'll pre-process, generate, reduce dimensions, and also make a UMAP. We'll do clustering and plot genes of interest, and also find top markers within the different clusters in the data set. So here are some of the basic questions that all of us have that are working for the first time on single cell data. So first is, how many good cells do I have? What does that mean? What do good cells look like? And we're going to look at that today when we follow along the markdown. So we filter for UMI counts, which is the unique molecular index, which is there for every cell barcode. Then we filter for doublets, which are like clumps of cells that are grouped together, which are not real cells, and checking for mitochondrial distribution as well. So whether you're working with whole cells or you're working with nuclei, it depends on whether you should even look at the mitochondrial distribution or not. So all of these questions are what we'll be addressing in today's session. And how many types of cells do I have? So we pre-process it, we reduce its dimension, we do all of the clustering, and then we see how many types of cells you have in the different clusters based on the expression. And then we find the identities of the cells, which are basically the top markers. And there are several ways to find top markers. If you know a lot about your data set, you can also see if the top markers that you get doing your analysis are the ones that you would expect reading literature. And if you're just exploring, then you're free to see how many top 25 or top 50 genes are expressed in each of the clusters. I think we have around 3 sections in this session. So I'll take a brief pause after each section and give you a chance to ask questions. Saskia is our TA for today. So if you want, you can also post questions in the chat and she'll try to answer that. And if we feel that, there are more questions or some questions that we should all discuss, then we can address it at the end of the session. Great. So let's get started. An estimated 16,644 children injured with guns in 2018. I'm going to share my Markdown. All right, so let's get started with the Markdown for today. I hope that the office hours session last week helped some of you getting things installed, and that you have R Studio and Monocle 3 installed for today's session. All these installations I try to do in the week prior to the session, because installations just take so much time that we don't have enough time left in the session to actually do any analysis. So, all right. So the structure of today's tutorial is in 3 parts. One is the main content that I will go through. The main content has 2 types of questions, bonus questions and a challenge question. So bonus questions are something that you all can attempt during the tutorial. And challenge questions are a little more difficult, and you'll probably need more information and more time to attempt them. So you are free to do those right now or do those when you go back. And then if you have any questions related to these challenge questions, you can always come back for office hours, and I'll be happy to help answer those. I've also, again, given you the links for the website and the GitHub and the Google group here so that you can try to look at it. Sometimes some questions that we have are already up there and already answered. So it sort of helps to have these resources up here in case you have any questions about a particular function within Monocle 3. All right. So today we'll need 3 packages. One is Monocle 3, one is ggplot2, and the third one is dplyr. So Monocle 3 is the one that needs maximum time for installation. So I asked you to install it prior to this session. If some of you already have these 2, that's great. You can just load them. But if you don't, then just follow these 2 lines first and install those packages. It should be super quick, and then just load the library in. So I'm going to get started with that. So here I'm loading Monocle 3. And then I'm loading ggplot2. And then dplyr. So all you need to do to load these packages is library and the name of the package once you have them installed already. All right. So a few preliminaries first. So the first thing on our agenda today is to know how to load a CDS object. So here I've already supplied the CDS object to you. We are using a human brain sample called the Sentinel. And we'll be reading that in today. So what I'm doing here is in the variable called CDS, I'm using the function read RDS, which is, so RDS format is a format in which the CDS objects are stored in a sort of a compressed manner. So read RDS, and I'm going to read in my Sentinel CDS using that function. All right, so that reads in my Sentinel sample. So I want to look at what I have read in. So I'm just going to do, I'm just going to type CDS, And this gives you a sort of summary-like information of what's inside that object. So it means that here it means that the class is cell dataset, which is it's a cell dataset object, and the dimensions are 27065 by 6979, which means these are the number of genes and these are the number of cells or nuclei. And then it also tells you what your row names are, what your column names are. So if you can see here, the row names are all the ensemble gene IDs, and the column names are all of the cell barcodes. All right, and so if you want to look further into what's inside the CDS object, then one of the things that you can do is head call data of CDS. So why do we do this? So CDS object can be viewed as an Excel sheet with multiple sheets inside it. So it's not a very straightforward column by row table. It's a more complicated table than that. So for that, you cannot directly access columns or rows. So the columns within a CDS can be accessed using an accessor function called call data, and the rows can be accessed by an accessor function called row data. So I want to see what my column data consists of. So I'm just doing a head, so it's going to display a few lines. So my call data for this CDS object contains the cell barcodes, which is the first column, the size factor, the number of UMIs per cell barcode. It also gives me a percentage of mitochondrial UMIs. And then I have a scrublet score and a scrublet call, which I'll talk about as we move ahead. Now I want to, so I've looked at the columns within my CDS. Now I want to look at the rows. Row data with the CDS. All right, so the rows have all the information related to the genes that are in your data. So if you see the first column right here is the ensemble gene IDs, but that doesn't give you any information because we don't really know what gene it is. So we also have a column for gene short name or the gene symbol that's right here. Since this is a human brain data set, you can see that all of the gene symbols are in all caps. And then the gene ID is here again, then the chromosome name, and BP1 and BP2 is just the start and the end position, and then what strand it is on. All right. And then head of counts in the CDS. So I want to see what kind of counts we have. So if you see here, it doesn't show you everything. There's like so many dots and then there's a number in between. This shows that the matrix is quite big. So it is stored in the form of a DGC matrix, which is a sparse matrix. That's how all of the single cell data is usually stored. That's the industry standard. So here we read in the CDS and we looked inside the CDS, we looked at its rows, we looked at its columns and saw what all is inside our CDS. Now, what if you didn't have this pre-made CDS to load in directly? And like, what do you do then? So there are two ways to load data or to make a CDS from scratch if you don't already have a CDS. And there are two different functions for it. So the first function is called the new cell dataset. This function is used when there is a repository or a website which already hosts a matrix, the cell annotations, and the gene annotations, which are the three files that are needed to make a CDS. So if the website or repository already hosts these files, then the only thing you'll have to do is download those files and then make a CDS. So for that, in that case, the new cell dataset function within Monocle 3 is very helpful. So here, all I'm doing is I'm reading in the URL from the repository for the expression data, the gene data, and the cell barcodes. And then I use the new cell dataset function to make a CDS. In this case, we won't have to use this function because we already have a pre-made CDS. The second function is the load data function. This is when you have the matrix files in the matrix market format, which I said is a sparse matrix and is stored as .mtx files. And so if you see all of the single cell studies that are being done right now and the data that has been submitted for it, one of the files that has been submitted for each sample is the matrix file, which is in the .mtx format. So to be able to directly process this file, if you've got it from, say, Geo, then you can use this load data function. All you'll have to do is give the path to where you downloaded the matrix file, give the path to the gene annotation file, and give the path to the cell barcodes file. If you give all these three paths and use the load data function, it directly makes a new CDS for you. So once you make a CDS, all the functions in Monocle 3 post making the CDS are exactly the same. So these are two functions that are very helpful, especially when you're trying to analyze already published data and you've downloaded it from some place and don't know how to get started. All right. So one more section here this is an extra note for 10x users, and there are more 10x users than other types of data, because the 10x has been used widely. So Monocle 3 has a function to read in 10x data so that you can do all of the subsequent analyzing steps just as you would for sky data or split-seq data or drop-seq data. So the function within Monocle 3 that can read in 10x data is called the load cell ranger data function. So all you have to do is make sure that you give the path to the 10x data correctly and it to the outs folder of the 10x data, which has the feature TSV file, the barcodes TSV file, as well as the matrix. So as long as it can find those three files from the 10x output, the load cell ranger function will do its job and make a CDS for you, after which you can again look inside the CDS using the same accessor functions. Just gonna take a 1 minute pause. Okay. All right, so I'm going to pause here a little bit and see if there are any questions at all about how to make a CDS, how to use these functions, how to load the data. This is pretty much the first step. So if anyone has questions, please go ahead and you can ask right now, or I can continue to the next part.
Hey, Aishwarya, I just had a quick question more about looking into the CDS object to look at the gene short names and what I think are the ensemble IDs.
Mhm.
So I feel like for humans like us, it's easier just to work with the gene short names. So why not just always work with gene short names? And is there a reason why there are, maybe one gene short name could have multiple ensemble IDs? I was just wondering more about like nomenclature for naming genes.
Yeah, of course. At the BBI, we map all of the data for collaborators to ensemble genes. Basically, the ensemble ID has a certain number of letters and then numbers, and then there is a dot and there is a number after the dot. So basically, the number after the dot only tells you what version of ensemble or what ensemble version gene version it is. So what we do is we just remove that part, because the last part, because basically it denotes the same gene. But we only give back the ensemble IDs because we're not sure what our collaborators want. But like you said, you can totally just use the gene symbol and do the analysis. Yeah, it's not mandatory to have the ID column as well.
Thanks.
I had a question about the like when you're loading in these cell data sets. So is there like a community standard for like the format of the metadata and the gene annotation files? Are they always, in a particular way, or do you actually have to format them before you put it into the CDS?
Right. So basically, the matrix file that has all your counts will have the cell barcodes. So the number of columns in your barcode file should be equal to the number of rows in your matrix file, which means that otherwise you're going to have a clash saying that the number of lines in this file don't match the number of, like, there are more cells in this and not enough in your matrix or something like that. So your number of rows and number of columns from gene and cell files, which are the TSV or text files, have to match the rows and columns within the matrix, which you can check also. And there is no standard for reading in the files. As long as those three files exist, you can make a CDS, and CDS is pretty much the input for most of the tools that are available for single cell analysis. Okay. Great. So I'll continue on to the next part. Okay, so now you have your data loaded. Now let's try some things. So as we discussed, we're going to first look at how many good cells we have. So what do good cells look like? They have reasonably high UMI, not too high and not too low. They have low mitochondrial rates, but of course, this is not a problem if you're working with nuclei. It only matters if you have whole cells that are going into the experiment. It also matters what tissue you're working with. So for example, we've noticed that heart tissue has more mitochondrial contamination than any other tissue that we've worked with in the past. And then good data also has singlets and no doublets. So we are going to try to filter for these three things, within our data set right now. So let's deal with UMI count filtering first. So if you looked at the call data of the CDS that you have right now, you saw that there is already a number of UMI column within the call data of the CDS. And when BBI returns data to our collaborators, we return the CDS to you with a default cutoff of 100. So any cell barcodes that have a UMI that is less than 100 are removed from the CDS and then returned to you. That is from the pre-made CDS. But if you want to make a CDS that has no cutoff done and you want to decide what cutoff you want to do, then we also provide the cell annotation, gene annotation, and the matrix files for you to make a CDS without any cutoffs and then apply your own cutoffs. So right now I want to apply a cutoff of 150. So basically I want to remove all the cells in this data set that have less than 150 UMI. So what I'm going to do for that is, and you can, so here what I'm doing is I'm taking my CDS and then I'm looking inside the call data of the CDS. And I'm going to the NUMI column and only keeping the ones that are above 150 UMI. So what I'm saying is within my CDS, take all the rows, as you know, in R, the structure is rows and columns. So I'm saying that within my CDS, take all the rows for which the column data has an NUMI greater than 150 only, and then put that output in my CDS again. So I'm overwriting my main CDS and making it a CDS that has only UMI greater than 150. But you are free to do something like an CDS and then UMI 150 or something like that, so that you don't overwrite on your CDS. Only that you will have many cell data set objects then, and then you have to remember which one is what. Okay, so I'm going to look inside my CDS again. So if you remember in the beginning, we had more than 6,000 cells, but when I removed cells that have less than 150 UMI, now I'm down to 5172. So you can see the drop in cells each time when you filter. And if you filter and there isn't too much drop, it means that your data is pretty good. All right. And a question here that I get very often is, this is a pre-made CDS that has been supplied to you and it already has the NUMI column. But what if you're working with data that has 3 files from which you made a CDS and there is no column inside the CDS at all? So what do you do then? How do you make an NUMI column? That's when you do this. So what you can do is you again do the CDS, and then what you're saying is, let me first add the column to the CDS. So what I'm going to do first is I'm going to take the call data of my current CDS where I want to add a new column, which is the number of UMIs per cell barcode. So what I'm going to do is I'm saying from the call data of my CDS, or in the call data of my CDS, I want to add a new column. I'm going to call it N UMIs. And then I'm going to use the matrix package, which is already within Monocle 3, and then use the column sums of my counts file, which is the matrix file, and then add it to the CDS. All right. So now if you do head of call data of the CDS, you should see a new column that's called NUMIs. And ideally, it should be the same as the column that we already gave you in the pre-made CDS. So this is how you add or do a column sum and find how many UMIs are there per cell barcode. All right. So what if you want to plot a distribution of the UMIs? So one thing that you should know, that within your CDS, whatever data you have present in the call data of the CDS, which is the columns within the CDS, you can plot. You can plot different plots like box plots, violin plots, UMAPs, anything that is within your call data can be plotted. You can make density plots or bar plots as well. So let's see, let's plot a qplot. And for this, we need ggplot, which we installed earlier. So I'm saying that I want to plot a quick plot for the call data of my CDS. And I want to plot the n.umi column. And I want to plot it as a density plot. And you get a quick plot on the right, but then that is very hard to decipher. So that's when you go to the log scale, where it sort of like spreads out better on your plot. And all you can do is just copy this again and say that you just want to take a log 10 of that column so that you can view it better and see how it spreads out. And you can see it better, the distribution. So like this, you can plot any of the columns within your call data. It doesn't always need to be the number of UMI column. And so within your data set, you can also have a very you can have cells that have unreasonably high UMI counts. And this usually can be an indication that your data has a lot of doublets, or there is leaking in the protocol, where because of which there's like extreme high UMI counts, which are outliers that are not falling within the normal range of what your data set should look like. So one of the things that you can do is you can summarize the number of UMI column to see what the 5-number distribution looks like, the mean, medium, and the quartiles, to see how far out the outliers are lying. So if your mean or median is somewhere around, let's say 1000, and you have the upper side at around 10,000 or like 50,000, that means that cell shouldn't be counted. And that could be like a couple of cells only, but it'll skew your distribution. So you just want to remove those couple of cells that have those unreasonably high UMI counts, because that's not real information. So you can do a summary of the call data within the CDS and a summary for the n.umi column. And then you can see that the minimum value is 151. And now you should know why, because if you scroll up here, we filtered for all the cells that have less than 150. We removed all of those cells. So your minimum value has to be more than 150. So that means our filtering worked. So the minimum value is just above 150. Your median is around 324. And you look at the max, it's like 40,000. And that could be a couple of cells only. So you can just remove that part. So all you have to do is, instead of doing less than sign, you have to do greater than. So basically you're saying that only keep the UMIs that are less than 40,000, let's say, or only keep the UMIs that are less than 39,000. So it'll remove these outliers here, and you'll have the confident data that resides right here within the mean, median, and the third quartile. All right, so let's do that. So I'm saying that I want to take my CDS, and within my CDS, I want to take all the rows for which the number of UMIs are less than, let's say, I want to say 30,000. Great. Okay. So let's look at the CDS dimensions now. So if you see, we looked at the CDS here. It's 5172 cells or nuclei, and it's 5171. That means it was only one cell or nucleus that actually had that incredibly high UMI count here, max, which we removed. So it gives a good, it gives you confidence that you're working with data that is falling within your mean, median, and third quartile range, which is this peak right here. So you're working with a good data to move forward. You're saying that I don't have very high and very low UMI counts. And now I'm going to filter for two more things. One is the mitochondrial reads, and second is doublets. So one of the things that you can do to filter for mitochondrial reads is just go to that column. So if you, for a refresher, let's look inside the CDS again. So there is a percentage mitochondrial UMIs column. So this is a percentage per cell barcode. So this is a data that has been generated from nuclei. So we don't have mitochondrial UMI contamination problem at all. And the industry standard, or what I have noticed from reading many papers, is that if you do work with whole cells and you want to filter for mitochondrial UMIs, then generally people use 10%. So keep all the data that has mitochondrial UMIs less than 10%. So here, I don't think we need to worry about that. So I'm not going to really do this because we didn't work with whole cells. And then the third filtering that we do is for doublets. So there are different, or like there are, so many doublet callers in use right now. The most famous ones are Scrublet, Doublet Finder, and we at BBI use Scrublet. The only reason is it's fairly confident and it is quick. We tried to implement Doublet Finder and it was glacially slow. So for NovaSeqs, it takes days just to process two samples through the Doublet Finder. So it wasn't really ideal for us. So we use Scrublet. And there is no industry standard to using one tool over the other. It's just personal preference. You just have to make sure that you're making the right calls when you filter doublets. And I will talk about that right now. So basically, when you call doublets using Scrublet, it simulates sort of a histogram, which is a bimodal. So it has a peak, then it has a valley in between, and then another peak. So the left side, or the left peak, corresponds to embedded doublets, which are generated when two cells have similar gene expression. And the right side is what the Scrublet group calls it neotypic doublets, which are made when cells with um, distinct gene expression get clumped together, or like with different cell types. So Scrublet can detect neotypic doublets because if you think about embedded doublets, cells or nuclei with similar gene expression are going to group together because that's how clustering works. So if there is a very, very apparent different cell type or a different cluster altogether that has been formed just comprising of doublets, that's what Scrublet is going to detect, because it's not a real cell type, it's just a doublet clump or a doublet blob in your UMAP. So that's what Scrublet is going to detect for you. So we run Scrublet in our pipeline to call doublets versus singlets. And then it also assigns a doublet score for each of your cell barcodes. So what that means is it's a score based on that bimodal distribution, and it'll tell you where to have a sort of cutoff to decide that left side of this is all my singlets and right side is all the data that is mostly doublets, and I'm going to throw it out. So that's what the bimodal distribution means. But as in everything that we do, things aren't so ideal most of the times, and you don't get a bimodal distribution. So that's when just a manual cutoff that the program does for you is not enough. That's when you look at the doublet scores and decide that, oh, maybe I cut off at 20%, or okay, I cut off at 15%. That completely depends on the data set, and it's different from data set to data set. So you have to do a few iterations to check what the right threshold for your data is. So it's a slow and steady sort of process, and it doesn't happen just right away. So that's when you use Scrublet scores to manually set a threshold and then plot the Scrublet calls onto a UMAP. And then you can see whether, are you seeing like stringiness between the clusters. So for we have noticed that for very large data sets coming off of NovaSeqs, which have, let's say like 30 clusters, there are sort of like these different clusters that are on the UMAP. And then if you closely look at the UMAP, there are sort of like these stringy bridges between the clusters, which are usually an indication that you have doublets in your data. But that shouldn't be your only reasoning to remove the doublets. You should also plot the singlet or doublet value on the UMAP, which we will do today, to check if there are actually doublets or not, or you're just removing good data. So yeah, it's a trial and error method. So if you look at the call data again, we have a column called the Scrublet score and a column called the Scrublet call. So the Scrublet call is the column that is made by using the threshold that has been chosen by the algorithm. So Scrublet chose a doublet threshold for us. And based on that, it said that these cells are singlets and these cells are doublets. And then Scrublet score is the column that you can use to manually set a threshold if you're not happy with how the calls are being made. So we provide both within the CDS for you so that you can make that call and that it's not being made for you. All right. So right now, what I'm going to do is I'm going to be very stringent and I'm going to ditch all the doublets in my data set. So what I'm going to do is run this line here. So what does this line mean? So I'm looking at my CDS and then I'm looking at the column called Scrublet call. So I'm saying that in the Scrublet call column, there should be no NAs. So NAs usually exist when there aren't enough cells, and that scrublet fails because of that, because it cannot run the algorithm because there aren't enough cells. So I'm saying that make sure that there are no NAs in the scrublet call, and then that all the scrublet calls are only singlets and keep only that data. So that's what I'm going to do here. And then I look at my CDS, and then if you look at the number again, I just lost one cell, which pretty much means that I don't have many doublets. You should also note that this is not a very big data set. So the chance of doublets being around aren't so high. Okay. All right. And then another thing, like I said, if you're not happy with how the calls are being made and you don't want the threshold to be set for you and you want to set it yourself, then all you have to do is run this part. So where you access the scrublet score column instead of the scrublet call column. So what you're saying here is within your CDS, take all the rows that have the column data scrublet score less than 20. So let's see. Okay. So now I set a manual score. So that means that the call column had a less stringent cutoff. And because of my more stringent cutoff of 20%, which is 0.2, I have lost around 1,000 cells, I think. Yeah. Yeah. Oh, 600 cells, let's say. All right. So that's how you manually set a scrublet score. Now a challenge question. There are two challenge questions in this part, after which I will take a break and let you ask questions. First is, in this data set that we gave to you, we already have a column that says percentage mitochondrial UMI, but that has been calculated by us and given to you. So how do you calculate this mitochondrial genome reads in your data and put it as a column in your call data is a challenge question. How do you know which genes are mitochondrial? So a hint is look at the row data and look at the chromosome column, and you should be able to pull out the mitochondrial chromosome and the data associated with it. And you should definitely do this because it helps for whole cells and then tissues. If you're working with heart, it's going to be a big decision for you to filter mitochondrial reads. The second question is, if you don't have a pre-made scrublet score and a scrublet call column, what do? How do you put these columns in your call data? So you have to run scrublet on the side and then use those output to put it inside as a column in your CDS. So these two are challenge questions that I highly recommend you try when you go back. And please come back with questions and I'll be super happy to help, because some of these early filtering is so important because sometimes after you do all of these filtering, you realize that your data there's nothing in there. So instead of doing all the analysis and then realizing that it's not giving any meaningful result, it's good to first do a very solid filtering. All right, so before we move to the next question, I'm gonna stop the share and then see if we have questions. Okay, so one of the questions that we have is the challenge question. So I'm not going to answer that, but that is a good question. Okay, does anyone have any other questions before I move ahead? Okay, so I'm gonna get started. Okay, so the next part is how many types of cells do I have? So the first thing that we will do is pre-process our CDS, which helps in normalizing the data and runs the principal component analysis. So let's run the pre-process CDS function. And now I'm going to run a function called plot PC variance explained. So, and I'll tell you in just a minute what that means. So if you look on the right at this figure, it tells you how many principal components or number of dimensions you need to choose while pre-processing your data, so that all of the variance in your data has been accounted for or has been explained. So the default for this number of dimensions is 50. So if you look at this plot here for our data set, which is the Sentinel sample, 50 is pretty much where the plot starts becoming horizontal, which means that almost all of the variance has been explained using 50 PCs or 50 number of dimensions. So at the moment, we should be good by just using pre-processed CDS directly. But if you have very large and complex data sets where this number is going up to say 100 or 150 or 200 even, so that means that you need to specify the 100 or 200 number of dimensions for all of the variability in your data to be explained before the plot starts becoming more horizontal or more stable. Also, if the plot becomes stable from 40 to 50, I would suggest taking 40 as your number of dimensions, because you don't want to overcompensate as well. So you wanna stop at 40. But if the plot is starting to become more and more flat at 50, then 50 is a good number. And the default is 50, so we should be good. And like I said, usually for NovaSeqs, we need to set a manual number of dimensions because it's a more complex data set.
Hey, Aishwarya, one question I have is like, is there a consequence for choosing 30? Like maybe would your conclusion be the same if you choose 30 versus you choose 50 versus you choose 20?
Yeah, so we have seen that the noisiness in the data like in the subsequent steps is more apparent when all of your variance is not explained. Yeah, so especially when you call top markers and you do differential expression genes, you want to make sure that all of your structured data variance has been explained in order to have confident top markers being called.
Awesome.
So yeah. So yeah, at 30, I would still be a little skeptical, but I would say 40 is still okay. But
so is it fair to say like consequence is greater for not including enough versus including too many?
yes.
Okay, cool.
Yes.
Thanks.
And by too many, I don't think you would want to, because once it starts becoming flat, it's pretty much like it's not needed. Because the program has done its job and your data is set to go to the next step. So if it's like super flat from 50 to 100, you don't need to have it specify 100.
Okay, awesome. Thank you.
Yeah. Okay. All right. So we've pre-processed the data and we've seen that our number of dimensions or the principal components are good enough to explain or account for all of the variance in our little sample here. So the next step is to reduce the dimensionality of the data. So for that, we use a function called reduce dimension. And if you don't specify any particular method, it uses PCA that it used for pre-processed CDS as well. If you're working with a TAC-seq data, for instance, instead of PCA, LSI method is better. It's a proven better method for sky attack or single cell attack-seq data. All right, so we've reduced the dimensionality of the data. Now let's run a function called detect genes and see what it does. So I want to see what this function exactly did. So I'm going to look at my call data and row data again. All right, so what detect genes did was add a new column in my call data. So it's telling me the number of genes that are expressed within this cell barcode. So that's pretty cool. So it says that like for this cell barcode, I have 3048 genes that are expressed. And for this, I just have 186 that are expressed. And what does it add to our row data? Let's see, head row data of CDS. So it adds a column in the row data as well, which shows number of cells that are expressed. So for this gene ID or gene symbol or gene short name, this gene is expressed in these many cells. So this gene right here, this ensemble 003, is expressed in 9 cells or 9 nuclei. So that sort of helps set thresholds. So if you're working with a kind of data set where you don't want to really include very, very lowly expressed genes, then these columns help for you to set a threshold. And then you can say that, using this column, I want to filter my CDS for all of the cells that have genes that are expressing less than 1 or 2, let's say. So one more round of filtering if you wish to do it. All right, and if you want to see in detail what a function does, you can also call the helper function, which is just a question mark and then detect genes. And then it'll tell you what the function actually does. And there's a good description here and what the usage is like and what the arguments actually denote. Okay. Now let's do plot cells. Plot cells CDS. Okay, so if you see on the right, we have a UMAP, but it is not colored. It's all grayed out because we haven't really clustered our cells yet. So the next main step is to cluster our cells. So for that, Monocle 3 has a function called cluster cells. And if I rerun plot cells now, you'll see that our data has 2 clusters, 1 and 2, that are colored differently. And one main thing that you can do here is add the cluster information to the call data of your CDS. So what that means is you want to know where each cell barcode resides, or which cell resides in which cluster, and you want to have that information as a column in your call data. So what you can do is make a new column in your call data called cluster, and then use the clusters function of the CDS. So what this does is head call data CDS. Okay, so if you see, it adds a new column in your call data that shows that this particular cell barcode is in cluster 1, whereas this particular cell barcode is in cluster 2. And this is super helpful, especially right now we have just 2 clusters, but it's very helpful when you have, say, 20 or 30 clusters and you want to know where each cell resides or comes from. Okay, so let's do a head of the number of partitions of the CDS. Okay, so what does this mean? It says levels 1 when I run this. So I want to see how many partitions my data has. And what that means is the number of clusters, but partitions are nothing but super clusters. So a group of 4 or 5 clusters may reside within a partition. So if you have big data sets where it shows that you have 30 clusters, you may still have, say, 10 partitions. And it could be that each partition has 3 to 4 clusters within it. So partition is nothing but a super cluster, and clusters that are similar in expression sort of get partitioned together. That's what it means. So here I have one partition, and within the one partition, as you can see, I have 2 clusters. So The same thing, instead of partitions, if I want to see how many clusters I have, instead of partitions, I'm just going to type clusters. Yeah, so you see, I have one partition and two clusters. So one super cluster within which there are two smaller clusters. So generally, when we work with B cells or T cells, what happens is when we plot it all together, there could be two main partitions and four clusters. So the two main partitions sort of group separately. One partition is usually T cell group, and the other partition is usually B cells. And then within the T cell group, there could be many more subtypes that are the different smaller clusters. I hope that puts it into more, makes it clearer. All right. Okay. So here it's just plotting cells and telling you that I want to, color it by partition. So it's coloring it by one color because there is only one partition. Now, if I want to color it by cluster, it's again going to color it by two colors because there are two clusters in this partition. So the plot cells functions pretty much helps you to plot all UMAPs in Monocle 3. It's a very straightforward function. All you have to say is plot cells, then you give input your CDS and just say what you want the UMAP to be colored by. And usually anything in your call data can be used as a value to color by. And we will do some of that plotting now. So as you know, this cluster column is in my call data. So I could use it to color my UMAP. Just like that, any other column in your call data can be used. So let's use the scrublet score column, let's say. Plot cells, I'm giving the input CDS, and then I'm saying color cells by scrublet score. Oh, scrub. Okay, so if you see, it's colored the UMAP by the column scrublet score in your call data. So as long as it's in your call data, go for it. You can plot anything. Let's plot the scrublet call. So ideally, since we filtered for all the doublets, all the possible doublets in our data set, it should all show as singlet. But if you do it prior to filtering for doublets, then you will be able to see doublets and singlets both. So it shows that all my data is comprised of singlets and that it's good to go. And yeah, you can also do a log of UMI column because UMI columns range is a lot. So you don't want the color to be like skewed. I mean, you don't want the values to be skewed and the color to be just like one all over the UMAP. So you'd want to do a log of the UMI column. So what I'm doing is, again, call data of the CDS. I'm accessing the CDS call data and saying that I want to make a new column called log.numi. And I'm doing a log 10 of the call data CDS UMI column. So if I do head of the call data to see if that worked, I have a new column here, if you can see, that is log NUMI. And now I'm going to plot that. So I want to color that on my UMAP. So log underscore n.umi. Yeah, so there you go. It shows you the NUMI column, and it shows the sort of the UMI distribution across your UMAP. And these types of plottings are very helpful when you're doing the initial filtering because, as I said, it's not a one-step process. Depending on your data set, you have to keep going back and forth to see what the best threshold is for each of these types of filtering. So using one threshold and then looking at the UMAP just to see how it looks like helps, and then going back if you want to change the threshold again, and then plotting and comparing the two plots. So, plotting call data values on the UMAP is a very great tool to have while doing filtering. All right. So the next step after plotting the columns in our CDS, why don't we plot some genes that I want to look at on the UMAP, the expression of the genes on the UMAP. So it's again the plot cells function. So you don't have to remember new functions for plotting expression of genes, which is a nice thing. And you just tell what genes you want to plot. So I'm saying that I want to plot some astrocytes because this is brain data. So I just give the name of some genes that I want to plot. And you can plot several. It's just that it's just going to get smaller and smaller and more compressed as you add more genes. So I would say if you think that some genes are working together as a module, then you might want to plot them together on separate UMAPs next to each other to see how they work in tandem or how they work in the reverse direction. So S100B. And then I want to plot just one other. Okay, so I'm using the plot cells function. My input is my CDS, and I'm saying that I want to plot these three genes on the UMAP, the expression of these three genes on the UMAP. Yeah, so it's going to show, like, see, these three genes are very lowly expressed throughout. There's some expression down here, but not much. So I'm going to plot some, let's say, neurons. Plot cells. Plot cells, input CDS, genes, and then RB-Fox 3, and then one more. You have 4. Okay, so this shows that there is some expression of RB-Fox3 right about here, and yeah, but still not very, very much. So maybe I don't have really good markers up here. And that's because I haven't really called any markers in my data set. So our next section is going to be calling top markers across our clusters. And once you call top markers or top marker genes, which are the topmost expressed genes across clusters, then you can come back to this step and take 5 top genes and plot it on your UMAP to see how the expression is across the clusters. Okay, so a bonus question here is, we use the cluster cells function to cluster our cells. And what if I don't like my clustering? I have a big data set and the clusters are all there. Suppose there are 15 clusters, but I don't really like the way they're grouping together. Like I have, suppose, T cells, and I have subtypes for my T cells, and they are sort of apart when I think that they should be together, at least within a partition. Then what do I do? How do I make that resolution better to recalculate the distance between the expressed cells and make them recluster and sort of make biological sense? That's when the resolution parameter comes into the picture. And you can try to run this with different resolution values to see if the clustering changes a bit. It doesn't change how, it doesn't completely change the underlying algorithm of how it clusters. It will just make better sense when it comes to cell type identification, when they need to cluster together as a group, even if there are several different subtypes within a cell type. So you should try this resolution parameter and check how there are slight differences in how the clustering works, and see if the top markers change at all. So a bunch of things you can try with each of the bonus questions as well. And if any of you work on human brain, you can also try plotting other genes of interest to you. I don't know much, frankly, about the human brain. So these are all the genes I have here, but you're free to try other genes. Uh, and then there is a challenge question here. So if you noticed, we have UMAPs on the right being plotted here, which is the default in Monocle 3. But you might want to check if the clustering is any different at all when you use DSNI versus UMAP. Now, bear in mind that uh, the biology behind the data will not change. So it is not going to look completely opposite, but you might want to check how different is it from UMAP. It should ideally be very, very similar, but just to try, like if you like t-SNEs better, these t-SNE plots better, then you can try using t-SNE instead of UMAP, which is the default. So you can look at plot cells functions and cluster cells functions and see what parameter to change it from UMAP to t-SNE plot. So yeah, you should try this as well. Okay, so I'm going to take a short break here before we move to the top marker section of our session, and look at the chat again if there are more questions. And we'll take just a 5-minute break and come back at 10:20. Okay, I'm gonna get started with the last part of our session, which is how to identify the identities of the different cell types in the data set. So if you're just exploring and you're completely new to the field, you can use the top markers function to determine which genes are, expressed or are specific to a particular cluster. And then use these to generate what's called a marker file or marker gene file, which can be used further for cell type annotation within the dataset. So to find top marker genes for each cluster, the monocle 3 has a function called top markers. So we're going to use the top marker functions capability to find us some genes that are expressed in our two clusters here. So I'm using the top markers and I'm giving the CDS as input. And I'm saying that for this small data set, I just want to see the top 10 genes that are highly expressed. And then I want to group cells by the cluster because I want to see the top 10 genes that are expressed between my two clusters in this particular data set. Okay. So this step, since this is a small sample, this is going to run very fast. But if you have really large data with gigabyte size of CDS object, then it's going to take a lot longer. And I would suggest doing it on the cluster. Otherwise, your local RStudio is going to crash for sure, which is not a fun thing because you have done so much hard work up until all the filtering and the plotting, and then it just crashes on you, and that's not fun. All right, so now we have run the top markers function. So we can look at this marker genes table. And that gives us a bunch of columns. So the first column is the gene ID, which is the ensemble gene ID. It gives you the gene short name. It tells you which cell group means cluster. So it tells you which cluster it is in. Then it also tells you the marker score. So how confident the score or the marker score is. So higher the marker score, higher the confidence with which you can say that that gene is expressed in that particular cluster. Then it gives you the mean expression. It gives you a p-value, a q-value, and a few other specificity markers, which gives you more confidence that the gene is expressed in that particular cluster, the top marker that has been thrown out by this function. So now that we have this table, what I want to do is I just want to take the top significant genes. So I'm using the subset function to subset out a few genes. So my input is the marker gene table, and I want to say that I want to use the marker test Q value result from that table and only keep genes that have less than 0.05 Q value. And then if I look at the top sig table, it will give me everything that has less than 0.05 Q value. So it'll only give me those entries. So these are my top significantly expressed genes across the two clusters. Now what I want to do is I want to find some top specific markers based on a specificity value from that table. So these specificity markers, like I said, tells you with more confidence that a gene is expressed in a particular cluster. So here I want to use the pseudo R2, which is a value that is calculated by the program or by the function top markers and added to that table. So I am saying that using that specificity marker, filter out the top one gene from each cluster. And since I have only 2 clusters here, it's going to show me only 2 genes, one from each cluster. So I'm going to run this part and I'm going to explain what each part means. So I'm saying that I want the output to go to top specific markers. I'm using the marker genes table as my input. From that table, I'm saying that only take the fraction of genes that are being expressed greater than 0.1 specificity marker. Group cells by the cluster. So I'm saying that I want to compare the top 1 expressed gene across the 2 clusters or between the 2 clusters. And I'm saying that I want only top 1. You can do however many. You can say I want the top 10 genes or the top 5 genes. I'm saying I just want top 1 between the 2 clusters. And I'm saying that use pseudo R2 specificity marker, which will tell me with more confidence that yes, this is the top 1 gene from this cluster. So when I do that, it gives me the top specific markers. So I want to pull out the gene short name for these specific markers, because if I just have the ensemble IDs, I'm not going to know what genes they are. So what I'm going to do is for these top specific markers, I want to pull out their gene short name and then find out what those 2 genes are. And now I want to see top specific marker IDs. So these 2 are my top specific genes. So I want to plot their expression as a bubble plot across the 2 clusters. So it will be the 2 genes and then across the 2 clusters and how much they're expressed. So what I'm going to do is plot genes by group, give my CDS as input, then top specific marker IDs, which are the 2 genes, and then group cells by cluster, and then ordering type is maximum on diagonal. So all this ordering type means that if you have a bunch of genes across a bunch of clusters, then the diagonal is going to have the maximum or the highest expressed genes being one after the other along the diagonal. And you'll see what I mean in a minute. And then just telling what the max size should be of the bubble. All right, so the 2 top expressed genes, which are along the rows here, and the 2 columns are cluster 1 and 2, because we have 2 clusters, and it's telling you how much it is expressed. So basically, Robo2 is more expressed in cluster 2 than cluster 1, and this HS3ST4 is more expressed again in cluster 2 than and barely expressed in cluster 1. And you can also use this function that we used before to plot the different genes. And now that we have the top 2 expressing genes right here, so what I'm going to do is I'm just going to put those names and see how it looks on my UMAP. So the 2 genes that we just found out are the top 2 expressed genes is Robo2 and HS3ST4. Okay, so that looks like it is quite nicely expressed, especially the Robo2. So this sort of like gives you confidence that your top markers have been identified correctly. So it would be nice to plot, like suppose if you do like top 5 genes, then you can plot all of these 5 genes as UMAPs here and see that your top markers have been called confidently. And I would still say that this is a small data set. In larger data sets, you can say with more confidence that it's been called perfectly. Cool. And the next part, or the last part, is to make a marker file. A marker file is very important because it helps you have an input. It's sort of an input along with the CDS for cell type annotation. And for that, we use the tool Garnet. After the next 2 Monocle 3 sessions on differential expression and trajectory analysis, we'll also have a session for cell type annotation using Garnet, which will sort of like take off on a third path, starting from making a marker, running your top marker function, making a marker file, and then doing your cell type annotation by classifying your cells first. All right. So how do you make that marker file that can be used as an input for Garnet? So there is a marker, making a marker file function called generate Garnet marker file. And all you have to do is give it the marker genes table that we got as a result of running top markers. And it gives you a Garnet marker file in the right format. You don't have to do anything in the exact format that Garnet requires, and it will get deposited in your current working directory. And I can show it to you, or we can look at it when we actually run Garnet in the future. But yeah, so this is how you find the top markers in your data set, check that the top markers that have been found are actually being expressed in your UMAP by plotting it on your UMAP using plot cells, and then make a marker file for further cell type annotation. Great. So these marker genes table is very, very helpful because it has so many columns with so much information that you can pull. And you can plot box plots, you can plot violin plots. You can also basically you can just export it as a CSV file and then do whatever plotting you want with it, because you already have ggplot in there. So it's a great way to dig more into your data. So marker files or marker genes table is all you need to do any kind of plotting of your top genes. So one of the tools that I have found is very helpful is Piano, and it will pretty much take the output from marker genes, not all columns, but like 3 columns from the marker genes table, and can be used to sort of see what pathways these genes are involved in, and do more gene ontology on it. So yeah, so this is pretty much the session, and I have some extra information here for what the different steps are, starting from QC and filtering all the way up to trajectory analysis, if you want to read through it and understand a little more of why we do each step. And with that, I will stop and take questions. And I already shared the slides with you and the markdown and the CDS object, but the recording also will be shared with you so that you can go over it if you weren't able to follow along.
Hey, Aishwarya, I have one question, though it might be better suited for your office hour potentially. So I'm actually really excited because I have sequencing data that just came off maybe like last week and a half ago. And so today was super helpful just to get oriented in what I'm about to do. But so it sounds like I need to run Cell Ranger first. But I'm also really eager to use the BBI pipeline so I can finally use like a really nice dashboard. And so does the pipeline and the dashboard incorporate Cell Ranger? Like, is it wrapping around Cell Ranger, or do I want to use Cell Ranger first? And where do I go to access the pipeline?
That's a good question. So our pipelines are open source. They're on GitHub, and I can share the links to the repositories with you. The pipeline is in 2 parts. One is the demux, and the second one starts with trimming FASTQs and ends with making a CDS. But the pipeline is very Sky specific.
Got it. Yeah.
So you would have to run Cell Ranger first, get the output from get the 10x output, And that output should already have the 3 files that are needed to make a CDS. So you don't have to run the pipeline part. You can directly start from Monocle 3.
Okay, awesome. Thank you so much. I'll check it out.
Yeah, and yeah, just Slack me or sign up for the office hours and we can talk more. Cool. And thanks everyone for signing up. This was great. And I will send out a feedback form. It would be very helpful if you could, do our survey. It helps us make future tutorials better. Yeah, thank you so much.